Assalamualaikum warahmatullahi wabarakatuh. Determination of microplastic in surface water and sediment of Kelantan Bay presented by Nur Ain binti Saiful Bahari from University Malaysia Kelantan, Jeli Campus. The introduction. The increasing rate of plastic production and usage of plastic globally is creating a hideous problem to the environment. In fact, plastic is strong, durable, and very slow to degrade, which can take up to 400 years to completely degrade. Plastics are polymers, which are a change of molecules that are derived from small molecules of monomers that are extracted from oil and gas. With time, plastics in the environment can fragment into smaller pieces of microscopic plastic particles, and when smaller than 5 mm in size are called microplastics. Microplastics in surface water and sediments for amounts, distribution, trends, and composition in marine environments have been proposed as one of 10 emerging issues in Malaysia, while in global context, Malaysia is among the top 10 worst polluters. Hence, it is important to understand what microplastic present in the marine environment, which potential to accumulate in the food chain. Therefore, the microplastic present in the surface water and sediment at Kelantan Bay were determined and characterized in this study. The sampling area. The sampling was performed in between May and July 2019. The Sri Tujuh Tumpat Kelantan was selected as a sampling site. The 3 sampling points at Sri Tujuh Tumpat were selected. Each of the sampling points was coordinated and illustrated as shown in picture and table. Next, we go through the methodology. Surface water analysis and sediment analysis. A water sampler was used to collect surface water samples. About 250 ml of surface water were collected and transferred into glass bottle. For the sediment sampling, an Ekman grab sampler was used to collect the sediment. The samples were transferred and stored in clean jar. At each sampling interval, the water sampler and Ekman grab sampler were cleaned carefully with deionized water to reduce cross-contamination. In order to avoid airborne or microplastic contamination, the jars were sealed with an aluminium foil before closing with their lids. After that, the surface water samples were pre-treated using wet peroxide oxidation method (WPO) in order to eliminate the interference of organisms in the sample. Meanwhile, for the sediment samples, the samples were first treated by drying method for one night or until completely dried at temperature of 90 degrees Celsius. Then, the samples undergo wet peroxide oxidation method. The continuous analysis for both sample surface water and sediments. Both samples underwent density separation process in order to determine the abundance of microplastic. After that, the mixture was filtered by using number 4 Whatman filter papers and vacuum pump. Then, sorting and identification of microplastic. All of the filter papers were inspected and photographed under microscope with 4 to 10 times magnification based on shape, color, and size. Lastly, FDI are being used to identify the functional group associated with polymer chemical properties. Types of microplastics found in surface water. As you can see in the table, the microplastic present in the surface water samples. For the sampling point number 1, the microplastic present was fragment and transparent in color, while for the sampling point 2, the microplastic present was also fragment and transparent in color, but the microplastic present in sampling point 3 was fiber and blue in color. Next, we go through to the types of microplastics found in sediment. The table shows the results of the microplastic present for the sediment. Sampling point 1, the microscope result. As you can see, the microplastic present was fiber and blue in color. For sampling point number 2, the microplastic present was fragment and red in color, while sampling point 3, the microplastic present was fragment and blue in color. The discussion. In this research, microplastics were found in the surface water and sediment sample at 3 sampling points from Sri Tujobich Tempat, Kelantan. The abundance of microplastic was high in point W3 and point S3 than compared to the other sampling points. This is understandable that point W3 and point S3 were located in the downstream which water flow to the sea from the river. FTIR results. There were 45 totals of microplastics that have been found using FTIR analysis. Based on this picture, the FTIR spectra shows 96.42% match with polypropylene wave numbers according to the library setup in the thermoscientific FTIR. What is polypropylene? Polypropylene is a tough, rigid and crystalline thermoplastic produced from propane or propylene monomer. It is a linear hydrocarbon resin. Why polypropylene? From the analysis conducted, most of the samples collected in Kelantan Bay contain polypropylene microplastics, which became the most abundant pollutant in the marine environment and bring harmful effects towards aquatic organisms. The presence of the polypropylene microplastics may be originated from the activities from a nearby fishing port in the proximity. The sources of the polypropylene could be from the residue of rope production and utilization of fishing gears and strapping that acquiescence to postulation. As conclusion, result of this study shows the presence of microplastics in variety of shape and color from both surface water and sediment of Kelantan Bay. The content of microplastic particles mostly in range of size 500 micrometer due to the depth of the sampling using the water sampler and also Edmond grad sampler. The deeper the sampling it was, the smaller the microplastic can be collected. Fragments or plastic films were dominating types of identified microplastics in the sampling sites. The characteristics of the microplastics show the different types of plastics found were very harmful to the living organism, especially aquatic life, as they can eat the plastic which they might think food and eventually die. Hence, this study will give the knowledge about the microplastic which need to be focused and prevented before posing a severe threat to the extinction of marine entity and disrupt the food chain that may lead to the hazardous bioaccumulation. Last but not least, the acknowledgement. The special appreciation goes to the Faculty of Earth Science UMK Jeli Campus. Our cordial appreciation to all laboratory staff, and this research was supported by FRGS 2019. With that, thank you and Assalamualaikum warahmatullahi wabarakatuh.